Inside Otzi's frozen body, scientists are looking for clues too small to see. If we only study the arrow, the wound, and the scans, we may miss the evidence that changed inside him after death. The false belief is that ice freezes a story in place. The reversal is that microbes can keep rewriting it. The Iceman did not just carry tools into the ice, he carried a hidden world. For decades, the famous story has pulled us toward the visible evidence: the wound, the weapons, the damaged body, the ancient murder mystery preserved in ice. Recent videos have returned to that story with a sharper edge, asking whether Otzi suffered a worse death than people once imagined. That question is powerful because it turns a prehistoric body into something familiar, a crime scene. But there is another question hiding underneath it. What if the smallest evidence is not extra detail? What if the microbial traces inside and around the Iceman are part of the real timeline, showing what happened before death, what changed after death, and what may have entered the story much later? That does not mean we can simply say living microbes survived inside Otzi. That claim needs careful evidence. Microbial DNA is not the same as living organisms. A dormant form is not the same as an active infection. A preserved trace is not the same as proof. But the possibility is still haunting, because if microbial clues can come from life, death, decay, ice, and modern handling, then the Iceman is not one frozen moment, he is a layered biological archive. And the hardest part is knowing which layer is speaking. A body like Otzi should have disappeared. Most human lives from around 5,000 years ago reach us as fragments: a tooth, a bone, a tool left in the ground, a trace of charcoal where a fire once burned. Soft tissue is usually the first thing the past takes back. Skin breaks down, organs collapse, clothing rots. The private evidence of a person's final hours is normally erased long before anyone can ask scientific questions. But Otzi was different. Found in ice, he survived not as a clean skeleton, but as a rare preserved human body. Skin remained, equipment remained, internal traces remained well enough for modern science to keep asking new questions. That does not mean the ice kept everything perfect. Preservation is not magic. Freezing can protect some evidence while damaging other evidence. It can slow decay but not erase every biological process. It can hold material in place but also expose it to meltwater, pressure, drying, minerals, and the surrounding environment. So the body is not a simple time capsule, it is a complicated archive. That is why Otzi still matters. Usually, ancient mysteries depend on things people built, carried, carved, or buried. In this case, the most astonishing object is the body itself. Otzi became part of a natural preservation system made from cold, exposure, decay, pressure, and time. The mountain did not simply hide him, it helped turn him into evidence. And that evidence is not limited to what we can see. Inside and around a preserved body may be digestive clues, immune traces, decay signals, fragments of microbial genetic material, possibly organisms that survived only in special forms. Some clues may belong to Otzi while he was alive, some may belong to the narrow window around death, some may belong to the long process after death, others may belong to the modern world that discovered and studied him. This is why the story needs discipline. Fact is not theory. Theory is not proof, speculation is not evidence, and in ancient microbial science, the most exciting claim is often the one that needs the strictest caution. A weapon can explain a wound, but can a microbe explain the timing of death? Most people meet Otzi through a crime scene: a man high in the mountains, a preserved body in the ice, injuries that invite a detective story, tools and weapons around him like evidence, scans that seem to pull a 5,000-year-old death into the present tense. That version is compelling because it gives the mystery a shape. There is a body, there is trauma, there is a final journey. There may have been conflict, pursuit, collapse, and a violent end. The familiar question is almost impossible to resist: who killed the Iceman? But the true crime version leaves something out. A human body is not only bones wearing evidence, it is a living system that, at death, begins becoming something else. Before death, it carries food, fluids, immune reactions, possible parasites or pathogens, and whole communities of microbes. Around death, the body's barriers begin to fail. After death, decay begins, even if cold slows it down. Later still, ice, meltwater, soil, recovery, storage, and scientific handling can all add their own traces. So the body is not just a victim, it is also a changing environment. A visible wound can point toward violence, but a microbial signal has to be placed in time. Some microbes, or microbial traces, may have been passengers from Otzi's life. They may reflect diet, health, ordinary gut communities, skin exposure, or the environment he moved through. Others may be connected to injury, stress, or tissue breakdown around the time of death. Others may be decomposers that became important only after life had ended, and some may not be ancient at all. They could come from ice, soil, meltwater, or modern contact after discovery. That makes the microbial mystery more difficult than the murder mystery. A wound asks what happened to the body. A microbe asks when it joined the body. This is also why dramatic scan-based stories have to be handled carefully. A scan can reveal damage. It can support interpretations about injury and stress. It can challenge old assumptions, but by itself it cannot turn every dramatic possibility into a proven scene. The same is true for microbes. Finding a possible pathogen does not automatically prove illness. Finding DNA does not prove a living organism survived. Finding something near a wound does not prove it was there when the wound was made. The smaller the clue, the bigger the caution. Before asking whether microbes can reveal the Iceman's final hours, scientists first have to ask a colder question. Which traces were his, which arrived later, and which may be misleading us? To understand that, we have to stop thinking of Otzi as a single object. He is a layered evidence machine. On the outside is skin that once touched clothing, air, stone, snow, and human hands. Near the injuries are places where the normal border of the body may have been broken. Inside are the stomach and gut, where traces from meals, digestion, health, and the body's own microbial world may have been preserved in some form. Around him were tools, clothing, ice, and the mountain environment. After discovery came another layer: recovery, storage, examination, conservation, and sampling. Each layer can carry microbes or microbial traces, but not all of those traces mean the same thing. A signal from the skin might tell us something about the living man, or it might tell us about the ice, or about later contact. A signal from the gut might reflect diet, disease, ordinary digestion, or slow changes that began after death. A trace near a wound might look dramatic, but drama is exactly where caution is needed most. Scientists can approach these questions in several ways. They can use microscopes to look for structures, residues, and patterns that suggest microbial activity or decay. In some cases, researchers may try to culture organisms, meaning they test whether material from a sample can grow under controlled conditions. But culture is difficult, and a failure to grow does not prove nothing was there. DNA analysis can be even more powerful because it can detect genetic fragments left behind by organisms that no longer exist as living cells. But that power brings another problem. DNA can survive after death. DNA can also be damaged, mixed, or introduced later. So the question is never simply, what did we find? The question is, where did it come from? When did it enter the story? And does it belong to Otzi at all? This is the central caution behind the entire microbial mystery. Finding microbial DNA is not the same as finding living microbes. A genetic fragment is not a living cell. A dormant spore, if present, is different from an actively living organism. A preserved community is different from a modern contaminant, and a possible signal is different from a conclusion. The popular phrase sounds simple: life inside the Iceman, but the science is more careful and more interesting than that. Some claims may point to preserved microbial communities, others may involve DNA traces, others may raise the possibility of viable organisms only if the evidence supports it. Those categories cannot be blended together without changing the meaning. That is why the body is so difficult to read. Ice may slow decay, but it does not give every clue a label. It can preserve ancient signals beside later signals, biological evidence beside environmental evidence, and real clues beside misleading ones. The Iceman carried a hidden world, but before that world can tell us anything about ancient death, we have to ask whether we are hearing the voice of life, the echo of death, or the noise of everything that touched him afterward. If one microbial signal comes from life and another comes from after death, then the body is not one timestamp, it is a timeline. That is the key shift in the Otzi story. The familiar mystery asks what struck him, where he fell, and how long he survived. Microbial evidence asks a quieter question: what kind of body was left behind for the ice to preserve? Start with the gut. During life, every human carries microbial communities connected to digestion, diet, immune history, and daily environment. In a preserved ancient body, traces from the stomach and intestines may help researchers think about what a person had eaten, how the digestive system was working, and whether the body showed signs of stress before death. This does not mean a microbe can casually reveal a final menu or diagnose a disease by itself. Ancient evidence is rarely that clean, but when microbial traces are compared with food remains, tissue condition, and other biological clues, they can become part of a larger pattern. Then there is illness. Some microbial material may point toward pathogens or toward the burden a person carried in life. But this is where the language has to stay careful. A possible pathogen is not the same as a proven cause of death. A trace of microbial DNA is not the same as an active infection at the fatal moment. It may suggest exposure, it may suggest colonization, it may reflect a disease process, or it may be too damaged, too mixed, or too uncertain to carry that much weight. The temptation is to turn every microscopic clue into a dramatic answer. The better question is what each clue can reasonably say. Wounds add another layer. A visible injury tells us the body was damaged. But microbes around a wound, if they can be confidently placed in the right layer, may complicate how that damage is interpreted. Some microbes may belong to the skin, some may enter tissue around injury, some may appear only after the body begins to break down. In modern forensic work, that difference can matter. In a 5,000-year-old body, it becomes much harder. The wound is not just a mark, it is a place where the body's inner world, the outside environment, and later decay could meet. And after death, the evidence changes again. Death is often spoken about like an instant, but biologically it is a transition. Circulation stops, temperature falls, oxygen levels change, cells begin to fail. Microbes that were once contained in the gut may shift. Other organisms associated with decay may become more important. If cold arrives quickly, some processes may slow. If the body is exposed before freezing, other changes may happen first. The body becomes a moving system even after the person is gone. This is the reversal. The microbes may not solve the murder. They may not point to a killer, a weapon, or a final sentence of suffering, but they may tell us what the murder left behind: a stressed body, a recently fed body, a wounded body, a cooling body, a decomposing body, and finally, a frozen artifact. That is the concrete takeaway. Microbes are not just background dirt. Interpreted cautiously, they may help separate life, death, decay, preservation, and modern contamination. That makes Otzi more than a frozen victim. He becomes a record of transition, not just a man who died, but a body that passed through stages, each leaving traces stacked inside the same preserved remains. The machine was the mountain, the archive was the body, and the most important microbial clue may not be the most sensational one. It may be the clue with the clearest place in the sequence. From life, from death, from decay, from preservation, from the modern world. Because once those layers are separated, Otzi stops being a frozen answer. He becomes something stranger and more useful, a timeline of ancient death. But the hardest part of that timeline is also the most uncomfortable. The wrong microbe in the wrong layer could turn a clue into a false confession. If microbes can help reconstruct ancient death, scientists first have to answer a brutal question: which microbes belonged to the man, and which belonged to everything that touched him after he died? Some microbial traces may come from Otzi's own body. They may reflect his gut, his skin, his mouth, his health, or the ordinary microbial world every living person carries. Other traces may come from the mountain environment around him. Ice is not an empty vault. Soil, meltwater, minerals, cold-adapted organisms, and microscopic particles can all become part of the preserved scene. And then there is the modern chapter: discovery, recovery, storage, conservation, study, sampling, the long history of human contact with an object that became world famous. None of this means the evidence is useless. It means the evidence has layers. Contamination is not an embarrassment in a case like this. It is part of the case. A preserved body is not a clean laboratory sample waiting untouched for thousands of years. It is a human body that lived in one microbial world, died in another, was sealed by a mountain environment, later entered the world of modern science. The task is not to pretend those layers are not there. The task is to separate them carefully enough that each signal is given the right meaning. That is why the most dramatic claims need the strictest controls. Finding microbial DNA is not the same as finding living organisms. Finding a possible pathogen is not the same as proving it caused illness or death. Finding a microbe near a wound is not the same as proving it was present when the wound was made. Each step has to be tested against simpler explanations: ancient background material, post-death decay, environmental mixing, modern contamination. This is where the recent fascination with Otzi science has to slow down. Videos can race toward the most shocking version of the story: a worse death, hidden suffering, living clues inside the Iceman. But the science is more careful than the headline. It asks whether a signal appears in the right place, whether it matches the expected layer, whether it can be compared with controls, whether the interpretation survives another possible explanation. That is not less mysterious, it is more mysterious, because the evidence is not just asking what happened to Otzi, it is asking what happened to the evidence itself. The Iceman did not just carry tools into the ice, he carried a hidden world, and after death, that world met other worlds: the world inside his body, the world of the mountain, the world of decay, the world of preservation, the world of modern science. Each one may have left a mark. So the question is no longer whether Otzi contains microbial clues. The question is which clues can survive scrutiny, and that is where the Iceman becomes something larger than a single ancient death. He reminds us that death in the deep past is rarely a clean scene. It is a process. A body does not simply stop and become evidence. It changes, it cools, it dries, it breaks down, it meets the environment. It is preserved unevenly. It is discovered by people who bring their own world into contact with it. Every ancient body is a negotiation between what happened then and what can still be read now. Otzi is famous because the visible evidence is so powerful: the face, the skin, the tools, the wound, the sense that a man from thousands of years ago has been pulled almost impossibly close to us. But the invisible evidence may be what keeps the story alive as science. Not alive in the simple sense, not a guaranteed world of living microbes waiting unchanged inside the ice, but alive as a question, alive as a method, alive as a warning that the smallest traces can carry enormous meaning if they are placed in the right layer. That is why the Iceman still matters. The murder mystery asks who struck him. The microbial mystery asks what the body did next. It asks what remained from life, what began after death, what the ice preserved, and what the modern world may have added. It asks us to look past the dramatic surface and read the body as a system. And in that system, the smallest clue may be the one most dangerous to ignore. If you want the next clue in this kind of ancient evidence trail, subscribe and follow the next episode. Because Otzi did not just preserve a murder scene, he preserved a hidden ecosystem, and somewhere inside that frozen archive, the past may still be leaving traces too small to see, but too important to dismiss.